Hi everyone. I hope you're ready to have a blast today. It's like a dad joke of science. I had to make it. It's a little bit cringe. Thanks for putting up with me. Today's lecture, we're going to learn about this tool that is called BLAST. It's a very, very popular tool used by both bioinformaticians and even biologists use this tool a lot. So oftentimes, if you think about it this way, you have a gene of interest and you want to identify other sequences that are similar to it. How would you go about doing it? Usually BLAST is going to be your first step towards that. So put yourself in the situation where you're working in a research lab and you've sequenced some new gene or really any kind of piece of sequence from an organism. Could be any organism, pick your favorite one. Could be birds, could be bacteria, plants, whatever it might be. And you're tasked with trying to find out, does a homolog exist of this piece of sequence that you now have in the entire Homo sapiens genome? We have learned about some sequence alignment tools in previous lectures. So one's called Needleman-Wunsch and one's called Smith-Waterman. The question here is, could we use those to solve this problem? See if this little piece of sequence that we have doesn't match anywhere in the entire Homo sapiens genome. Well, if we remember, Needleman-Wunsch is used for global alignment. So that would be taking this little piece of sequence and trying to align it across the entire the entire Homo sapiens genome, the entire human genome. So it would be a whole bunch of gaps in that. So likely we couldn't use Needleman-Wunsch for that. Smith-Waterman is a local sequence alignment tool. So it's going to take that piece of sequence we have and try to find the piece of sequence in the human genome where it would potentially could match. So maybe we could use Smith-Waterman. But one thing we haven't talked about yet that adds complexity to this is something called algorithmic complexity. I know, I used complexity twice in the same sentence. I'm so sorry. But algorithmic complexity is something that is very important in more of the computer science side of bioinformatics. So you know how I've mentioned before that bioinformaticians oftentimes have to put on their biology hat to make sure that the results are making sense? Bioinformaticians also need to be putting on their computer science hat sometimes to make sure that these tools, these algorithms that they're using are going to be the most efficient way to come to our answer. And so algorithm complexity is a whole study within computer science where it takes a look at, given the steps that we have in this algorithm, and when I say algorithm, I don't know if we've actually talked about what an algorithm is yet in this class. So think about it like a recipe. So like you need to do XYZ thing to make a cake. In this case, we're saying we need to do XYZ thing to identify if there's a homolog to this sequence. So those XYZ steps have a specific amount of time that's going to be associated with them. And usually we can figure out what that time would be based on how big that input sequence is that we're either using to search the genome or based on how big the sequence we're trying to search is. So in this case, a whole genome. And so what we try to do in computer science and then in bioinformatics when we're doing it with these biological algorithms is we try to say, okay, given our sequence size or given whatever we're putting into the step of recipes, how long would this likely take the computer? If you're not familiar with running things on the computer that can take a really, really long time, and probably you aren't, that's totally fine. You're probably used to just like clicking on a button and at most it takes like 30 seconds for something to load. But if we're not really careful in bioinformatics and in computer science in general, things can take days, years, months to actually run. So this whole idea of algorithm complexity is what helps us decide, given our size of our sequence, is this actually going to be a reasonable thing for us to use to find this solution? And so both Needleman-Wunsch and Smith-Waterman good to use when you have relatively short sequences. So when you're using things like we've done so far in this class, like one gene, it'll work pretty reasonably well. It likely ran pretty fast for you when we were doing things in the lab, in the exercises, etc. We have some examples of actual timings within the slides. So less than 0.04 seconds to align two sequences that are 1,000 base pairs long, and 3.82 seconds to align two sequences 10,000 base pairs long. So, you know, it's doing quite well. That would probably feel like it didn't actually take that long. It wouldn't feel like Windows booting up on our machine and you just get that circle that goes forever and ever and ever. It seems reasonably well. But the runtime for both of these algorithms is directly related to the square of the sequence length. So just like in math, when we have a square, let me just draw this here. So like 2 squared would be equal to 4. Adobe, not helpful. This just means you're taking whatever this number is and multiplying it by itself. So to generalize it, if we're talking about sequence length, we'd usually give it some sort of variable like we would in math, where it would be something like n squared, so that you could just plug in whatever that is. In this case, it could be 1,000 base pairs and multiply it by that. And that would give us our runtime in general. And so depending on your compute resources, it could be around 0.04, but it's generally going to be about the same for most computers that we would use. So every time we increase the length by 10 times, so that would be this example right here, 1,000 to 10,000, we're going to increase the runtime by 100 times, because remember, it is squared. So if you do 10 squared, that is 100, which is why we see this here. If you look at this example, when we're doing 1,000 base pairs, it's 0.04 seconds. When we multiply this by 10 to get 10,000 base pairs, we multiply the runtime actually by about 100. So if we took 0.04, multiplied it by 100, it would be 4 seconds, 3.82 seconds, pretty close. So there's always going to be a little bit of a wiggle room there, but it's not perfect, I'll say this, in terms of being perfectly 4 seconds, but it's in that ballpark. Whole chromosomes can be huge. And that's just one chromosome. That's what, 100 million base pairs in length? That's already going up so much from this example where we had 1,000 base pairs. So still, imagine, go back to this example that we're in, where we're a researcher in a lab and we're taking this sequence that we have and trying to compare it to this huge genome. Well, that huge genome squared is going to be a whole bunch of runtime. So if we took the entire set of human genes, that's around 20,000 to 30,000. We can say 30,000 worst case, 20,000 best case. And then we have our new gene, and we use Smith-Waterman, our local alignment tool, which we thought we could probably get a reasonable answer with that. And just even look not even at the whole genome, but just each set of human genes against X one at a time. What would that timing be? And I won't make you do the math. We did the math for you. You are welcome. But each alignment with this gene, this mystery gene, if it took 5 seconds, it would take 42 hours to do all of the alignments. Imagine how inefficient that would be. We sequence new chunks of information in research labs, so many of them every day, that we need to do these kinds of investigations for. If we had to do 42 hours every time we sequence some new information, it would slow down the research process so significantly. Just imagine if we didn't know it was related, wanted a homolog, say in humans, we wanted to search everything that we had in the entire GenBank database. That's 240 million sequences. Remember, this was just 20,000 to 30,000 sequences would take 42 hours. All of GenBank would be 34 years. Imagine doing like a master's degree and be like, sorry, you need to do a sequence alignment here, so you're going to be stuck in your master's degree for 34 years. No, it's not reasonable. Not at all. It's wild. So we needed a new solution that might not give us the best answer, but would give us a reasonable answer. So one thing to remember with Smith-Waterman and Needleman-Wunsch sequence alignments is that they waste a lot of time by trying to find the best solution, the perfect solution. There is no better solution than this solution, which is great for certain use cases, but it does not scale well for tasks that require multiple pairwise alignments, like this example that we had previously. They're just too slow. I'm sure y'all have seen on the internet machine that triangle where it says you can have something fast, you can have something cheap, and you can have something correct, but you can only pick two. So, same kind of idea here. If you want it correct and cheap, then it's not going to be fast. And almost all bioinformatics things are cheap because we usually want to open up our tools that we're building to other researchers as well. So if we want something fast and cheap, it's likely not going to be correct or perfect, but it could be good enough. So there's this trade-off that we have. It is a very frequent trade-off that we see in computer science where we're looking at the difference between accuracy and speed. It just takes a lot of time for the computer to go through and figure out what is the actual best. Same with humans. If we need to go through and do all of those pairwise alignments and do them absolutely perfectly, we're going to have to double check, triple check our work to make sure we didn't add any errors. And it's just going to take us longer. If we can be happy with a pretty good job where we don't need to double check and triple check and all of these things, we'll get that done quicker. Same idea with computers. If they can say, we're making some reasonable guesstimates here that are going to lead us to a very good solution, but it might not be the best, they're going to be able to do that a lot quicker. And so this is where BLAST comes in. BLAST is not going to give us the perfect answer, but it's going to give us a reasonable answer a lot quicker. It's one way to think of it is like the Google of biological research. So when you go onto Google, you're typing in some keywords. Google doesn't guarantee that it's going to give you the perfect response back in its hits. It gives you a whole collection of hits that we can kind of go through and say, yes, this makes sense. No, this doesn't make sense. I'm going to click on that and read that article. I'm not going to bother with these other ones. BLAST is the same kind of idea. It goes through, it does its best to search through key sequences. We'll talk about what those kinds of things are in a minute, to figure out what are the most likely hits. Same as a Google results page that would come up. What are those most likely hits that then the bioinformatician or the biologist can go through and say, yes, that makes sense. No, that doesn't make sense. We're going to dig into this one a little bit more, whatever that might be. So BLAST stands for Basic Local Alignment Search Tool. I will always just call it BLAST because why not? One thing bioinformaticians are great at is making hilarious acronyms, so you're welcome. We might be bad at UI, but we're great at acronyms. BLAST was created in 1990 at NCBI, and its main purpose was to compare protein or nucleotide sequences to various sequences in the NCBI databases. Similarly to Smith-Waterman, it's going to make local sequence alignments, but that was a weird word. Local sequence alignments. But in this case, instead of us providing that other sequence that it was getting the local sequence alignment like we would have in Smith-Waterman, BLAST just searches all of the sequences that are in a database. So there's some terminology, of course, sorry, that we have to get used to when we're talking about BLAST. The sequence that we submit is called the query sequence. It's what we're using to query or search that database. BLAST is then going to perform a bunch of pairwise alignments against the entire database. And in this case, the entire database is the target. So all of those sequences in that database are what the query will be targeting. BLAST will then report local alignments that they think best match our query sequence. Even though it's performing local alignments, one thing to note is it's not using Smith-Waterman to do those alignments because that would still take too long. Remember the 34 years example? We don't got time for that. So we will get into the details of what it's actually doing in some future slides here. There's no guarantee that BLAST is reporting the best matches. So remember our triangle? We're going for fast and cheap here, fast and cheap. But it is going to do its best to get us a reasonable answer. The local alignments it reports involving sequences aren't going to necessarily be the best. It will result in faster answers. It is doing everything it can to give us reasonable answers. So they might not be correct, but they should be at least reasonable. it has become the primary method of database searching and constructing local alignments in all of bioinformatics. So very, very popular tool. Why would we use BLAST? So there's a whole bunch of reasons that you might find yourself thinking, oh, hey, BLAST could be a good solution for this kind of problem. So a common one is to determine potential orthologs or paralogs for either protein or nucleic acid sequences. We could be using it to determine what proteins or genes are present in a particular organism, determine identity, so what's similar, what kinds of how many nucleotides are similar or how many amino acids are similar in a DNA or protein sequence. We can use it to discover new genes, determine variations of genes, determine amino acids that are really important for functions or structures of a protein. So there's lots of different reasons that we might be using it. The BLAST website is on the slide here. You could also Google NCBI BLAST and it will pull you up there. You could also have BLAST locally installed on a computer or a server if you want even more advanced options. We won't be using that in this course, but just know it does exist and some research labs do utilize that. So there's lots of different variants of BLAST. I'll go over them briefly here and we'll see examples of them in our assignments and later on in the lecture as well. So we have BLAST-P, which is BLAST protein, if you think of it that way, is where you're taking a protein query sequence, which remember that's the one you're searching the database with, versus the protein target database. So the target is the whole database, all of those sequences in that database. So it's a protein sequence, searching a protein sequence database. BLAST-N or BLAST nucleotide is going to be a nucleotide sequence, query sequence, versus a nucleotide sequence database, which would be the target. BLAST-X is a nucleotide query sequence, so we're passing it into the nucleotide, that will get translated in all reading frames. Reading frames. Why am I having such a hard time with my words today? I'm so sorry. And then those translations will get searched in a protein target database. So it's actually the translations that are the query, so not the nucleotide itself. T-BLAST-N is a protein query sequence versus a nucleotide target sequence database that has been translated in all reading frames. So you might be thinking, why would we want to even do this? There are situations that will come up where we would want to do these different things, where we're taking a nucleotide sequence and we want to identify potential proteins. That can be really helpful for identifying homologs. But we'll get into those examples later on. And then the final one is a T-BLAST-X, which we take a nucleotide sequence translated in all reading frames versus a nucleotide database that is translated in all reading frames. So you'll see here, these top two, BLAST-P and BLAST-N, are probably the ones we will use most often and are used most often. The rest of these options here, so you'll see once we start doing translated stuff, we start seeing T's here. BLAST-X, so X is going to be translated in all reading frames, and you'll see it when we have this one down here and it says T-BLAST-X. Again, we're doing it versus a nucleotide database translated in all reading frames. So, it's something to get used to. It takes a while to learn all of these different meanings, something to try to just put into the memory of like, hey, these are the different things that exist. Should I need to use those for a specific biological question I have? For every BLAST search that we do, we have to pick which variant we want to use. So from this list here, we have to pick which one do we want to use. The computer won't know. We have to tell it. We then also need to specify a query sequence, and I'll show you how to do that in an example coming up. We can either do an accession number, which is really nice, or we can paste in the FASTA contents. We have to select which database we would like to search and which program we would like to use. So there's a couple different variants of BLAST that we could use, and then some optional parameters. We will go through all of this in an example. There's also some more reading available on the course website if you would like that, to dive deeper into some of these different parameters that we're going to talk about, as well as the outputs that we would get. So selecting our databases. So our default database that we would like to use is the non-redundant one. It's going to include sequences from GenBank, Protein Databank, SwissProt, and others. but it collapses all identical sequences together. So this is going to save us a little bit of time in the search because we're not going through duplicate sequences and then getting those duplicate sequences in our results. We can also restrict the sequences sequences to only be RefSec sequences or any of the other databases that we talked about in topic 2 of the class. For BLAST-P and BLAST-N, we have the option of restricting to a specific organism if we were only interested in one organism or even a set of organisms. Again, I will show you how to do this in an example coming up, but we can also exclude a specific organism or a set of organisms. So say we knew our sequence came from Homo sapiens and we wanted to find homologs in other species. We could actually restrict BLAST so that it didn't search any of the Homo sapiens sequences, which is really nice. Oops. Some other parameters that we have, which these words won't really mean much, like the word size isn't going to mean anything to you yet, but we'll get into what that means. But things like our mismatch scores that we talked about with our regular sequence alignments, but that is like the worst way to describe it. Our Needleman-Wunsch and our Smith-Waterman sequence alignments. We had our mismatch scores, our gap costs, etc. We can set those, but they can also be automatically adjusted depending on what program we select. And we'll see all of those later on here. So the very first thing we're going to do is we're going to perform a blast of the sequence NP_000221 versus the NR database. So first and foremost, I'm going to show you what the BLAST website actually looks like. Let me just scroll to where that is. Here we go. There. So when you click on BLAST or you Google NCBI BLAST, this should be what you actually see when you load it up. So you'll see it says Basic Local Alignment Search Tool, which is BLAST. It gives you some idea of what it does, any updates, and then if we wanted to download it to our machine, we could here. But again, we won't be doing that. So here it shows some of the main ones that we might use. So our nucleotide, our protein, our BLAST-X, our T-BLAST-N. And if we wanted to start it off, so remember our very first sequence that we were going to search a BLAST for was a protein sequence. And the reason I know it was a protein sequence is because the accession number started with NP000221. So the NP is a protein RefSec accession number. It's okay if you don't remember that. We did go over it in an earlier class. It takes a while to remember those things. But also if we click on the link that was in the slides, it should take us to this here. And we can see here, if we scroll all the way down to our sequence, that it actually is a protein sequence down here. So we have two options for what we can do in BLAST. We could be doing a nucleotide blast, which wouldn't make sense given the fact that we're using a protein sequence. We could do this translated nucleotide, it doesn't make much sense. We could do a protein to a translated nucleotide, which in this case, we're just doing a very initial blast, so that probably isn't the way we want to go. Or we could take our protein sequence and search it against the protein databases, which is probably where we want. So I'm going to click on that one there. Here, we can either enter the accession number directly, which was NP_000221, or if you wanted to instead, if we just go over to the NCBI page again at the top, where we had that sequence, we can click up in the top left corner, go to FASTA, and we can copy this FASTA here and we can paste it in. I'm going to only use the accession number for this one because I want to show you that it works the same. If you want to take the sequence on your own page, do it, and then you can be convinced that it actually does work as expected. So you'll see up here when we are in our actual BLAST-P that we have these tabs at the top which show all of the different BLAST alternatives that we have. The one highlighted is the one we're in, and that's the one that we want. We also see it up here that it says standard protein blast, which again is the one we want. We enter our query sequences, which remember those are the sequences we're searching the databases with up in this top first box up here. I just put the accession number, but you could copy paste the sequence or the whole FASTA sequence if you wanted as well. When you put the accession number in, it should automatically fill out this job title, but you can also change this to be whatever you want. I find it really helpful to make this descriptive because it will actually save your search after you do it. So if you needed to come back and look at it, say for an assignment, if you made this like A3Q whatever, it'd be really easy to identify what you were looking for. Now in this question, we're asked to search the NR database. So as we continue to scroll down here, we'll see our databases. We have our standard database, which is NR, or our experimental database. We want the radio button selected that says standard, which it should be automatically. And then we can confirm that by looking down here underneath the standard, and it says non-redundant protein sequences, which is what we want. You'll see underneath that, if we click this drop down, there's also all sorts of other options we could select. So for instance, if we're ever asked to use the RefSec proteins, then we would be able to go into that and click that instead. But in this case, we're using the non-redundant protein sequences. Right here is where we can filter it to either be specific organisms or to exclude specific organisms. Words are hard today. We're going to leave that blank because we weren't asked to do any of that. And then down here in our our program selection, we just want our regular BLAST P. We don't need to worry about any of these modifications to the algorithm. If you wanted to learn more, you can always click that question mark and it will bring up the help page. So it'll give you a little bit of an idea of what that is, but you can also scroll all the way up to the top, click on this help up in the top right corner, and it will give you more information. It's very, very well documented. So if we knew for sure that was all we wanted to change, all we have to do here is hit blast. And then what it's going to do is it brings up this job page. It gives you your request ID. So if you accidentally exited out, you would have it. It also tells you it's currently searching, when you submitted it, and what the current time is, and the time since submission. So remember, this is searching a ridiculous number of sequences. It is going to take more, usually more than a few seconds here to actually identify what it needs. The BLAST algorithms are really interesting, especially for the course, because if everybody's working on their assignment at the same time, say in a lab, it will take longer to get those BLAST results. But that's just because we're asking a lot of the computer at the same time. Once it runs, it should pop up a window that looks something like this. So again, we have our job title up at the top. If you wanted to see the search summary, this is going to give you all of the details of the different parameters, which are kind of like the specific ingredients that we plopped into the algorithm. So all of those are listed up here. All of the things that it searched, like look at the number of sequences that it searched. That is wild to me. Like, oh, so many. It would have taken so long if we were using Smith-Waterman. I would be talking for 34 years and we wouldn't want to hear that. Anyways, so where we really want to see our results, and what we do is we scroll down here. We see as we're scrolling, we had the BLAST-P program, we did the NR database. This was our query ID that we had. This was the description it pulled from the database. It was an amino acid. It had that many amino acids in it. And then this is where our results actually are. So we'll see here, there's different tabs that we can click on. We're going to start here with just the descriptions tab. So the descriptions tab is going to be sorted by the hit that they think it's most similar to. And you'll see in this case, they think our sequence is most similar to this leptin precursor from Homo sapiens, which, if we go back to the NCBI page, we actually did submit a sequence for the leptin precursor to Homo sapiens. So it actually found the sequence that we submitted, which was great because that should be in that database. We have all of these different values over on the side here, like our scores, our query cover. So it's 100% of our sequences represented. This thing called our E-value, which is going to be really important. We'll talk about that later. And our percent identity in this case is 100%. So that means how many amino acids are identical. And you'll see here that if we click on this, it would actually take us exactly to where in the results it has the two sequences that it identified. So the top one, the query here, is the sequence we submitted. And the bottom one is the sequence it found in the database. And so the middle line here is the alignment between the two of them. So in this case, because it's 100% match, that is going to be identical. If we scroll down to some of our other hits, I'll find one where we have more differences here. Here's an example. You'll see that we have a plus in our middle sequence. That's because those two amino acids aren't the same. So you'll also see here that our identity score is not identical because there's actually two pluses. So we don't have 100% identity for this one. Whoops, opening Copilot by accident. We have 98% identity. So that's kind of what that looks like. But I'll scroll back up to the other example that we were at. And so this was the one we were at here. It has all of that same information that we saw on the other page, just that information that we were given here. And so up here too, we see our sequence ID that we submitted. There's other information like other titles that it could have, etc., etc. If you want to go back to the descriptions, you can either click on the descriptions tab up here, or you can go over to this right hand side and just click that descriptions and it will take you right back to where you were. The tab that we were on when we clicked this that showed that was alignments. So if I click alignments and I scroll, you'll see this is the exact same tab that we were on. I'm just going to go back to descriptions now. Over here, we can see after the description where we clicked and saw the alignment, we then get the organism of interest. We could click that. It's going to take us to the taxonomy browser. Maybe not the most useful thing for us. It opens a new tab though, which is nice. So to get back to where we were, we just have to go back to the previous tab. And then over on the side here, it shows you the accession number of the sequence that it pulled from the database. And so if we click that, it will pull up NCBI, and it's actually the exact same accession number as the one that we searched. So that's all the information. If we were trying to say, hey, what are the homologs? We're probably not going to include this top hit as a homolog because it's the actual identical sequence. We would then go down one and look at the next organism that isn't Homo sapiens, which in this case is Pan troglodytes. And we have a totally new accession number that we could click on to find more information, different percent identity. And if we click on this, it would give us all the details for that, which is quite nice. So just back to our BLAST page, the other tabs that we haven't seen yet. So this graphical summary, it will show you for the query sequence that you have up at the top here, how well do each of these hits align over the whole length of that? So are they the whole length, which is these red ones up here, or are they partial? You'll see some that don't have the front up at the top, and you can click on any of those and it will actually tell you what was the score, and you can go to the alignment and find more details there. So there's lots of clicking around that we can do within BLAST. So if you wanted to see one of these ones here that were not maybe as aligned, you can go in and it would pull you up. Oh, I think I scrolled your actual sequence. It would show you exactly what portion aligned, and you'll see that it didn't have the full length of the sequence aligned, which was 167. And you could take a look into that in more detail. I'm just gonna keep scrolling up here to go back to the top. I think I could just hit back and it would work as well, but that's okay. And then the last tab that we haven't explored yet, this, I should have just hit back, is taxonomy. And so this is going in to show you the number of hits from the different organisms that you can see here. I don't personally use this tab very often, but it does exist if we ever needed to pull some information from there. The other thing we can do, and we'll use this in a future example, is we can filter the results for only a specific organism or group of organisms or excluding organisms, as well as filtering based on some of those key metrics that we get in those results. So here we go. Let's just go back to the slides here. I think we have another example coming up, but that's okay. So the BLAST variant we used was BLAST-P because we had a protein sequence and we wanted to search the protein database. And the most similar hit was actually the one, the protein sequence that we inputted as our query sequence because that was contained in that database. So now we're going to do another BLAST search with that same accession number on the NR database, but we're going to restrict the organisms to wolf. And we'll look at how these results are different. So here I'm just going to go back into BLAST. Let me open that up. And what I want to do is I'm going to go up to the top here, and in the top left side I can say edit search. And we have the same information that we have here. And the only thing I'm going to change is underneath the choose search section and the standard database where it says organism, I'm going to start typing, which one is it? Yes, there we go. The organism name that we want to restrict. So we wanted Canis lupus, which is taxid ID 9612. As you start typing it, you should get this little list pop up. You can just kind of search through, click the one you want, and we want it to only be that one. So we're not going to be excluding it, which is what we would do if we wanted it to not be included in the search results. We would click that little button. We just want it to be only that organism. So everything else can be left the same. Let's hit blast. It will think about what it's doing. Great option to have a cup of coffee while we wait. I laugh, but I probably always have coffee while I do blast searches. Again, it will take some time to identify. Could take longer, could take less time. Depends on how many requests are being sent into the server at the same time. Our results page looks really, really similar. So again, all of our details for what was inputted, as well as up to here, all of those ingredients, and you'll see the number of sequences that we searched was far fewer than what we had in that original search. And if we scroll down, we only have 4 hits. Wild compared to what we saw before. So you'll see that all of these hits are from that one organism that we specified, and that they have some different values here for percent identity than what we would have seen before. So the reason we didn't see them on the main hit page for the previous one is because these percent identities were much lower than options that they had for other ones. If we go into our graphic summary, so you'll see here again, there are a couple that go across the whole length of the sequence, and there are some that don't. And you see these different colors down here. That's related, to the alignment score. And the legend for that is just up in this top left corner here. So I want to click onto this first one here. I want to see our alignment. So this is what our alignment looks like. You'll see we have our query sequence up at the top, which would be that one from Homo sapiens, and then the target sequence that it found from the database up at the bottom. And in between here are the consensus amino acids. So we see we actually have some gaps. Those gaps are when the 2 amino acids aren't in the same amino acid family. The pluses are when the 2 amino acids are different, but in the same amino acid family. So they're a little bit more conserved. And that's why we see things like this positive score with our protein alignments, where it will be slightly higher typically than our identity score. Our identity score is how many are identical, how many amino acids are identical in the 2 alignments. And the positives are the number of amino acids that are identical or in the same amino acid family. So that's why sometimes you see that be a little bit higher. But yeah, very, very similar to what we saw before. Feel free to look around on that a little bit more. I don't want to talk through everything again. I feel like you can just rewind the video if you want to see that again instead of having me blabber on about BLAST. All right. So with BLAST, we have 2 kinds of scores that we will see. We have something called the raw score, which is going to be calculated based on the scoring matrice that was used in our search. Very, very similar to what we did when we were covering sequence alignments in a previous lecture, and we were actually adding up all the matches, the mismatches, the gaps, the extensions, etc. The thing about raw scores is they cannot be compared directly with different BLAST searches because we might have used different scoring matrices for those different BLAST searches, so we can't compare them directly. Bit scores are based on our raw score, so they're related to our sequence alignment score, but they're going to be normalized based on the parameters that we used for the BLAST search. So they can be compared directly to identify which alignment or which target in our database is a better fit for our query sequence. So oftentimes we'll be using bit scores to compare between BLAST searches, and then raw scores we would not use to compare between BLAST searches. Before we jump into this slide of this example, and I'm gonna just read from it once we're into the BLAST again, But I'm just going to show you here the example of the different, if I go over to descriptions, the different scores. So we have our max score, or sorry, as I goof up all my things, our raw scores and our bit scores. Let me find where this is. Here is our raw score in our brackets. It takes me always a while to find where that is because we literally never use it in bioinformatics for things, but I do know we test you on it. So it's good to know where it is. The raw score here is in the brackets when you're on the alignment page. The bit score is the one that comes first and is followed by bits. So this score here can be used to compare, for instance, between this search and the one we did in exercise 1, whereas this score here, the raw score, can't. It's related to the parameters. So you'll see they're slightly different. Bit scores are normalized. We use them to compare. Raw scores are just the raw score from the sequence alignment. That should be that. All right, so I'm going to go back to BLAST. Now, our next exercise we have is another protein sequence, NP_05913, and we want to search it versus the NR database excluding Homo sapiens and using the BLOSSOM 45 matrix. So what I'm going to do is I know that's a protein again because it's that NP accession number, which is a RefSec protein accession number. I'm just going to paste in, that accession number. Again, on the slides, you can click in, you can search it in the NCBI database, you can get the FASTA sequence, but you can also just do this and it makes it so much easier. If you didn't see the job title, it gets auto-populated once you click somewhere else on the page after you've pasted that in. We want it to search it against the NR database, which we have here. You'll see that here I have still the dog genome, sorry, not the dog genome, the wolf genome still selected. That's just because BLAST was trying to be helpful. And remember the, the settings I had. Oftentimes when you'll restart one, you might want to hit restart page up top here just to clear any filters that you had from previously. We are then going to exclude Homo sapiens. So how we do that is we go back into this organism section and we start typing Homo sapiens. One thing I will show you, which is hilarious to me as someone who always types badly. There are the typos of Homo sapiens also, but they all link back to the same taxid ID, which is the actual Homo sapiens one. So you'll see there's many a typo here. Pick the one, any of them are fine, as long as they go back to this taxid ID of 9606. And then because we wanted to exclude them, we click this exclude button right at the end of that. And so that should remove any of those organisms, the sequences from those organisms being included in our hits at the end. Then the last thing we wanted to do was use the BLOSSOM 45 matrix. And so if we're looking at this page, we might not see where that is. What we need to do is, before we hit this BLAST button, expand this algorithm parameters. So remember, this is like the ingredients that we're passing in. This is just like more specific ingredients. So we're saying, oh, use this kind of butter instead of this kind of butter because it makes a difference, right? So down here, all we need to do is go to the scoring parameters. And right now the default is BLOSSOM 62. We can change that to BLOSSOM 45 right there. It's going to say, hi, you made a change. Make sure you want to make this change. We highlighted it in yellow and we added a little symbol. We did want to make that change. And then you can either hit the BLAST up here to start it or the one down here. Either or works. The same buttons do the same thing. And again, it will bring up this and it will start searching. While we wait for this to go, I'm going to start another BLAST for the second part of the question. And so again, it wanted us to do the same accession number, the same database, excluding Homo sapiens, but using the BLOSSOM 90 matrix instead. So again, we're going to go into protein BLAST. I'm going to paste in that accession number, click somewhere else for it to auto-populate that job title. I might actually add to the front of this one, BLOSSOM, that's spelled wrong. I just made a joke about typos and then I spelled things wrong. Blossom 90, because that will be the one we use. You'll see that all of my settings from the last time are saved. So we still have the NR database. We still have the Homo sapiens excluded. And if I go to my algorithm parameters, we still see we have Blossom 45. Let's change that to Blossom 90 and then hit blast. So this will let this run while we take a look, hopefully, at the results for this soon. We'll see how quickly they go. So this one's been running for 1 minute and 13. This one's been running for 9 seconds. I probably shouldn't have submitted them both at the same time when I want the results semi-quickly, but that's okay. We'll see how this all goes. So what we're going to be looking at once we see these results is we're going to look at seeing how are they different using Blossom 45 and Blossom 90. And I guess a quick gut check for y'all is, what is the difference between Blossom 45 and Blossom 90? Which one is used for more distance? Which one is used for more recent ancestors when we look at evolution? And the other thing we'll look at is filtering out these things called synthetic constructs. So those are very common when it comes to doing research work. They are literally just synthetic constructs that come out of some of our research work. There's lots of those that are included in the databases that we don't actually want to look at. We'll see. If this doesn't go soon, I might just let it keep running and then we can go over we'll come back to this and we'll go over what BLAST is actually doing while we wait for it. So how does BLAST actually work? So I mentioned this a little bit before, that it worked kind of similar to Google in terms of keywords. But the keywords in a BLAST search are actually bits of our sequence. So when we put in our query sequence, it's going to be divided into words of a fixed length. Typically, this is 6 base pairs for proteins and 28 base pairs for nucleotides. For each word that we would have in that sequence, we rank the similarity to all other possible words of the same length. Now, the convenient thing about this is that we as bioinformaticians, know that we can calculate this ahead of time, store it in a database to look up, and that it won't change. So we're able to effectively just have a lookup to figure out what are the similarity for all these other words. And then we identify what we would call high scoring words based on some sort of threshold. And that threshold we can change in those advanced algorithm parameters, but we usually will just use it as default. So all high scoring words, so words that matched up with a different words, chunks of nucleotides that match up with another chunk of nucleotide that is greater than some threshold, would be a high scoring word. So for each of those high scoring words, we find all occurrences of those words in the database that we're searching. These are called seeds. So they're effectively seeding our search. Once we have those seeds in the database, we will start extending the alignment in each direction until it falls below some sort of scoring threshold based on the alignment score, which is usually called X. And again, we can change that in our advanced parameters, but we usually just leave it as default. At the end, the alignments that get reported to us are all of the ones that are above some other threshold A. So we have three thresholds that we're working with. We have the reporting threshold A, we have the extension threshold X, and I'm just scrolling through, and then we also have the high scoring word threshold T. So those are all different numbers potentially that we can tweak and change as needed for our example, but we'll in this class typically use the defaults. But those thresholds are what lets us make those educated guesses on things to have them go quicker. So think about it in terms of the extension threshold. Once it goes below that threshold, we just stop. That's not to say that if we would have kept extending it, like we would see in, say, Smith-Waterman type of local alignment, that it wouldn't go back up. But what we're saying in BLAST is, for the sake of time, this is good enough. Once it drops below this, we'll just use that and we'll report the results of that if they're over the threshold A. So that's why we can't guarantee that we get the best result out of it, but we usually get a pretty good result out of it. So there's all sorts of advanced parameters that go in with which database we're selecting in sections. So where we're saying like Blossom 45 or Blossom 90, and we can tweak the word size, which I mentioned. So the smaller the word size, the better it's going to be at finding more distant homologies, but it's going to take longer because we're going to have more seeds. It's going to result in more alignments getting extended out, etc. When we go, and I'll show you this when we go back into to BLAST here, but the nucleotide BLAST has 3 different program selections, and those are actually just modifying the word size. So the default one that we use is MegaBlast, which is a word size of 28, but then there's also discontiguous MegaBlast, which has a word size of 11, and Blast N, which also has a word size of 11, but there's slight differences between the last 2. So I'll let you read that on the slide. I don't feel like I need to read that out loud. We also have an option to filter low complexity regions, again, in that same section where we would choose which scoring matrix that we wanted. So filtering low complexity regions removes or ignores sequences that don't have a lot of context. So these are going to be things like repeat regions. So dinucleotide repeats are when just 2 nucleotides repeat over and over and over again. Those don't necessarily have a lot of information in them. They can also be regions that are usually kind of different in length, so they don't provide a lot of value for our sequence alignments. All right, let me see. Do we have our results? We have our results. So before we get into e-values, let's switch back over to here. So this is the very first search that we did. It has Blossom 45 as a matrix. We can double check that if we want to come back to it by clicking search summary up here, and we'll see which matrix was used. And then if we scroll down to our descriptions, you'll see these are kind of the results that we get. So we got a couple high, I think these are all kind of like the monkey ape type of ones. 1 synthetic construct coming up in there. Gorillas, okay, these all seem kind of related to what I know about the evolutionary history of humans. And remember, we shouldn't see any Homo sapiens in here because we restricted those from being included in our results. So that was with Blossom 45. With Blossom 90, if we scroll down, you'll see that our first hit is a synthetic construct. So that's different than what we saw over here. And you'll see if we compare things like our query cover, our percent identity, those are going to be slightly different, and that's okay. You'll also see that we do have some difference in the ordering that we get in all of this. So the last thing that we were going to do is we were going to filter out the synthetic constructs. So how we filter those results is if we go up into this gray box up in the top right corner, and then you can just start typing exactly what you want for the organism, and we want this synthetic construct taxid ID 32630. We click that and we click exclude, because if we just put that in and then put filter, it would only show synthetic constructs. But if we hit exclude and then we hit filter, that should remove that very first one that we saw up at the top. I'm going to do the same thing for our search that we had with Blossom 45. So again, up in that gray box in the top right corner, start typing synthetic construct, select that, hit exclude, click filter, scroll down, and we'll see no more synthetic constructs as well. Now for the sake of comparison, let's just compare the top 2 hits. So this first one here that we have for Blossom 45 is from Pan Troglodytes. It has a 99% query cover, 99.32% identity, and then that's the accession we get from it. If we go over to Blossom 90, we have Pancensis. Oh, the word's already hard today, and now I'm pronouncing scientific names on YouTube. This is just too much. That's okay. We have a 99% query cover and a 99.42% identity. If we click on here to go look at the alignments, we can actually get the bit scores so that we can see which one is likely a better alignment. So this one here has a bit score of 2,189. And this one here has a bit score of 2,202. So this would indicate that actually the Blossom 90 was a better matrix for identifying homologous genes in other organisms in this case, because we have a higher bit score. Again, we can't use the score that's in the brackets because that is dependent on what parameters that we set for our search. And you'll see if we looked at the score, it would look like we should be using this one instead, but we cannot compare with just the score. We have to use the bit score. If you have any questions about that, please plop them into the course discussion board. The next thing that we're going to be talking about here is this thing called e-value. So you'll see right now, e-value is the one that's highlighted that's showing and ranking our results here. So e-value is a way that we can measure, statistically speaking, how likely it is that we would have saw this result just by random chance come up in our Google search of our biological database. And we're going to get into the slides here to talk more about that. But that e-value is what gives us the statistical confidence that what we found is actually legitimate and not just random chance. So let's pop over back into the slides here. Here we go. So e-values are how we can tell how good our results are. They're based on statistics, and the key point with an e-value is that we want to ensure our hits, our Google hits, our results are not likely to just occur by random. We have a lot of sequences in these databases. It is possible that we could just by random chance run into something that looked similar to what we have. The size of these target databases that we are searching strongly influence this random chance that we're talking about. When we're trying to identify homology, it is extra important to have confidence in the hits that we see not being there just by random chance, because we need to be crystal clear. I wanted to say 100% clear, but it's like, that's not reasonable. That's just me being over dramatic in terms of statistics here. But we want to have some level of confidence that it's just not random chance that two things look similar. And that's why we have to rely on these e-values and statistics. So an e-value is what we call actually an expected value, which might help remembering what it is. And it's just a quantitative measure of whether a returned alignment or a hit from the database represents a significant match or whether it could have occurred by chance. So you remember back from those examples, those e-values we saw up there were 0.0, and that will be important as we get into this here. So the lower the e-value, the more confident we are that the hit was not due to chance. So the lower the e-value, that means the more likely the hit is due to homology, which is what we're often after in these searches. The lowest possible e-value is 0, and the higher the alignment score that we have, the lower the e-value that we'll see. So the 0 means that it was given that database of that size, there is a 0% chance that this hit just happened randomly. So say we return a blast, or we submit a blast for a sequence, and we get some score S and an e-value of 1. This is going to be the reverse of the case that we just talked about. This means that in a database of the size that we searched, we are likely to see one match with a similar score of S to occur by random chance. So when we're saying random chance, that means without some sort of evolutionary relationship or without some sort of homology. Oftentimes in research, we will consider an e-value significant for homology as long as it's below 10 to the negative 5 or 10 to the negative 6. So that would be 0.00001 and 0.000001, I believe, if I'm doing that math correct. You can just throw that into a calculator if you want to see the decimal places, but that's what we're looking for. If you want more reading, you can take a look at those sections of the textbooks that are in the library. But again, all of the information that you'll need for this class, we went over in these slides. But of course, if you have questions, throw them in the chat or the discussion board. The chat. What am I, a Twitch streamer? And I will be happy to help you there. All right. Have fun. Have a blast, should I say. You'll be doing this in an upcoming assignment, and it will be great fun. We'll talk to y'all later. Bye.